The human genome was done about the year 2000, so now we have the end of the beginning. So here we are almost 20 years later, and now we're in the era of synthetic genomes, okay? So the next 20 years, we'll be probably seeing synthetic organisms. I'm not sure they'll release them into the population. Maybe we don't want a zombie apocalypse, but at least we'll potentially have new types of living organisms to do, the concept is productive things, okay? Cleaning up oil slicks, getting rid of pollution, that kind of thing. And here's the DNA, and I know people have studied that from high school up. Here it's the Watson-Crick hydrogen bonds, 3 versus 2, GC, AT, exons, introns, so gene splicing. Chromosomes, we have 23 chromosomes. You can have an X sex chromosome or a Y. So males are typically XX, females XX, males XY. If you've had genetics, there are XOs, there are XYYs, and some other altered genetics here. Of course, trisomy 23 is Down syndrome. There's Edwardian syndrome, which is too many 18s, and there's some other rare human genetic diseases here at the chromosome level. This is a chromosomal karyotype. These are all our pairs lined up. These are metaphase chromosomes. They're condensed, and they've been stained with histological stains. And the black stains are the tightly wrapped areas, and the lighter areas are the euchromatin. So there's heterochromatin, euchromatin, and you probably hit this all in genetics here. Notice the amount of chromosomes are different. Humans have 46, but sheep have 54. So there's not a complexity concept. Horses have 64. Okay, so carp have 104, chickens have 78. So you can't quite correlate chromosomes with sophisticated organisms necessarily. DNA sequence ATGC, here it is. This is the sequence. If you go to GenBank and look at a protein's DNA, they will actually give you the exact DNA sequence for every protein in the database here. And you can actually start with the DNA, go to the GenBank, okay, GenBank, and you can get the protein. Yay. And you could find the amino acid sequence there, the DNA to the protein. Here's deoxyribonucleic acid for DNA, sugar phosphate backbone, anti-parallel, hydrogen bonds, genes. You've done all that. There it is. It's what a nucleotide is. It's the bases, the sugar, and the phosphate. A lot of energy in the sugar phosphate backbone. And here's some naming. We won't worry about the names necessarily. Certainly in biochemistry, I think you have all the names. Here's the deoxyribonucleic acid. So this is a polymer making the sugar phosphate backbone. Here's the DNA, the hydrogen bonds, the base pairing. And here's the DNA sequence that can be coded. So if you put in a DNA sequence, software can actually put it together, find the protein information, and spit out the protein sequence, the predicted protein sequence. So if we have somebody with a disease, if you sequenced somebody with a disease with all their DNA, you can actually then look at all their proteins, and you can compare to wild type and find protein or proteins that are mutated, and that can give you a head start into understanding a disease. Okay. RNA is ribonucleic acid here. You have a uracil instead of a thymine. And so you've done it, and it's single-stranded. Okay. Genetic information from DNA to proteins. So the messenger RNA, so that should be messenger RNA here, not just RNA, because there's ribosomal RNA, tRNA, small nuclear RNA, what else? siRNA, there's pxRNA, oh my goodness, PXI, I think. There are a lot of different RNA, very complex. People think it's simple, it's not. Okay, there's a lot of complexity to the RNA here. Messenger RNA, ribosomal RNA, talked about that. There are some viruses that are actually double-stranded RNA molecules. So proteins are enzymes that change the substrate's bonding and potentially cut a substrate into multiple uh, uh, pieces, products. They're signal transduction, they're transporters, they form structure, and they regulate processes. So this slide refers back to the slide probably 20 or so ago, which showed the function in the middle and all these different activities. So certainly, you know, you should probably already knew all this, but if not, you definitely need to understand what proteins are at this point. Different amino acids, we've talked about that. Some proteins are small, some are large. Okay. Bacteria has about that many. Humans, I think it's closer to 20,000, I think, with the human genome, but at one time, this was probably information before the human genome. Not sure it's 30,000. Here's the triplets that code for the different amino acids in biochemistry. Medical vet didn't, you'll have to memorize all this. Yay. And eventually you can get a protein sequence, and those are usually one letter. So in the protein database, this actually is the code for a different protein here. Okay. So it's an alphabet, one letter alphabet there, and that's the code there. Proteins have different R groups, and they're trying to show you in this one chart these overlying R groups with the one letter codes here, and finding out which ones have a charge, a positive charge, a negative charge, hydrophobic, okay, hydrophilic, small and tiny, and polar, non-polar, lots of different properties there. And that's how you start understanding the three-dimensional structure of proteins. And again, we've covered protein primary, secondary, tertiary, and quaternary structure. So this is kind of a review of where we were previously. Here's just a cartoon showing the different levels of structure here in proteins. And again, we've hit this. That's why I'm being a little jaunty here through this. So DNA determines, gene sequence determines protein sequence, protein determines the structure, protein structure determines how it folds, how it functions, what proteins it binds to. So protein partners, PPs, protein partners, to make the molecular machines that do a process, M&Ms. Here it is, the central dogma, it's called. Okay, you've seen that before. Here's transcription. You've had this, how the DNA is unwound, and there's a leading in, well, that's the replication. This is RNA polymerase, and you end up getting, and that should be messenger RNA, not just RNA here. And that is then going on to make the proteins here, the proteins. Okay, so there's actually RNA polymerase have pockets, and enzymes itself is an enzyme, ACE, ASE. And here's the direction going down the gene. Okay, and there's a stop signal at some point. TAA is one. And there are proteins that tell the train, if you think of this as a train, to stop. So it halts it, and then it lets everything go, and the messenger RNA is released. And then there are proteins that bind to the messenger RNA and serve as chaperones that then go out the nucleus, the nuclear pore, okay? And so you have to go out the nuclear pore, you have to have the right papers to get out, right zip code, and then you go to the ribosomes here. Here's the ribosomes and the messenger RNA, and then there's the little protein, look like a string of pearls with the different amino acids coming out. Okay, so you can imagine that. So gene expression is known as transcription, turning on and off. Transcription can also be small ribosomal RNA genes, and these are non-coding or small RNA that code RNA that's not translated into a protein, tRNA or ribosomal RNA. Here's translation. You see the proteins being built on the ribosome, the A and the P site. The codon, the anticodon, the tRNA carries then the amino acid on one side with the anticodon on the other. There are enzymes that find the tRNA and load them. There's amino acid synthesis. Imagine you have the right tRNA but the wrong amino acid. So we could actually program a cell to make different proteins based on the same sequence. So we can make our own mutants, just changing the whole concept of the translational process there. People have done that. Here's the A and the P site. There it is, A site, P site. And I know in micro you did this. Here's the feeding, and here's the methionine, valine, glycine, alanine, leucine. So there it is. And then that's the peptide bonds being formed, and the new one is bonded to the methionine. It's a new bond, and that keeps going, okay, in a cycle into a stop codon. And then if there's a stop codon, there's no tRNA available, and there's kind of like it idles, the machinery's sitting there, and then it breaks apart. They're still working out exactly all the machinery that tells it to stop, okay? But that's kind of the concepts here. And then here's our friend, the tRNA. Look, it's a piece of RNA that has a lot of secondary folds and structure here. Okay, here's where the amino acids go and are loaded. Okay, and there's the anticodon up here. Okay, so the anticodon and the so-called amino acid acceptor arm here kind of looks like this structure, kind of a boomerang structure there. Okay, the anticodon. Here, the amino acid is stuck, so it's bonded here until you get into the previous A site, and then the machinery clips that bond, and that is then a peptide bond to the protein here. Here's the one letter, the triplet, the so-called genetic code. I know in genetics and micro, you definitely talk about the genetic code. And a gene supposedly codes for at least one protein. There is alternative splicing, okay, splicing, where a gene can give multiple messages, multiple messenger RNAs, because they've been differentially spliced. And so you can end up getting proteins. Alternative splicing. So that's kind of interesting. You have one gene that can give multiple proteins. Viruses actually do that a lot as well. Viruses will create giant proteins called polyproteins, and then host enzymes will break the polyprotein into the individual virus working proteins. So a virus can actually use host activity here to make multiple proteins from one giant protein. But in this case, this is a gene codes for two or more, that's my two, messenger RNAs, and you get proteins. And you're saying, when does that happen? Signals, stress, signals could be stress, infection, viral infection can change the splicing patterns. Okay. And so we end up, and this is where bioinformatics really allows us to look at metabolic networks. This is the next level inside the cell physiology, and also given a name metabolomics. Dr. Grace, if you're curious, he's interested in metabolomics in tomato plants, I believe. So each one of these boxes represents different enzymes and proteins. Those arrows represent interactions. The lines with a little kind of straight line, kind of a parallel thing, that means it inhibits. So they have isolated all these proteins in a protein database. So actually, there is software that allows you to draw these databases and show all the interconnections. So when I said top down, you were probably thinking 2 or 3 proteins. To me, top down means everything in the cell, we start there. So in a disease situation, and by the way, this is just cell cycle. What you see is probably not even all of it. How cells divide. These are all the players, or some of the main players in how cells go through G1, S, G2, and mitosis and back again. And it's not showing the signals. It hints at the signals, like DNA damage. If a cell has received radiation damage, then there is information that will probably shut down DNA replication if the DNA is broken or damaged. Cell growth factors come in this part. Here's a growth factor here. There's a signaling pathway, and you end up pushing the cells into dividing. So cells, when they come out of G1 mitosis, they're small. They have to get big enough by the G2, so when they divide by cytokinesis, that the cells are big enough to survive that, right? If the cell did not ever grow, it would be divided. You'd have little wee cells that you wouldn't have cells. Okay, something would be missing. So cells use G1, S, and G2 to replicate the DNA and get their mitochondria, ribosomes, everything set the table to go into mitosis. And this are all the proteins that have been involved so far. So this is a protein network guide to cell division, living cells here. So this is the real world, and this is what it is. So people go into medical, pharmacy, vet, this is the level they're going to expect you to be at. So that's why I'm starting a little slow, but you know, I got to be real. This is why this course is challenging, because you're used to going, you know, on a tricycle, and now here we are on a Harley, rocking and rolling. So get ready. But again, we will cover this kind of level, and I will go from this level down and back up again depending on what we're studying. But just wanted to kind of get you ready for a good taste of metabolic networks there. And this just reminds you about the so we were just here, okay, but we had been in this region here, and we'll be moving up or down again. And bioinformatics and the software allows us to make these trips up and down this organizational pyramid here. So that's we have help now. Biologists have help from in silico biologists, and this is what they're helping us do, is move from one level to the other and back again, especially looking at wild type versus diseases and whatnot. Okay, so that's my 10-cent tour of the first chapter. Kind of a lot of it's been review. If it wasn't review, you probably need to go back and hit your notes. Probably to make it through here, you should have done very well in microgenetics, is probably highly suggested, but you should have already known that. And we'll just rock and roll here. So hopefully I'm going to post this. It's Saturday today, so we'll see what happens. Okay, see you next time. Are we having fun yet? Hang in there. Study hard. You can do it. Woo woo.